and have a good discussion. We do want this to be discussion based, so definitely chime in. And then when you do ask a question, feel free to introduce yourself, introduce where you're coming from and maybe your general background so we can get a feel um, for where you're coming from with your question. But when you're not asking a question, if you don't mind to keep your mic muted and to avoid interrupting each other because we want to facilitate a great discussion because we only have 1 hour. And then with that, totally fine to disagree, totally welcomed. We're talking about some interesting and new topics, so certainly disagreement can happen, but don't forget to be kind when you're doing it. And there are no bad questions, so definitely ask all kinds of questions. The area of microplastics was totally new to me, so I know I have questions, and but let's try not to get stuck in the weeds, given the time constraints. And with that, we'll go ahead and jump right into contextualizing the article. But first, we want to welcome Dr. Matthew Campen and also Dr. Garcia with us today. If you're welcome to introduce yourselves so that we can get to know you throughout the conversation. But they're the authors of this awesome paper that we chose for the discussion for this group coming from the University of New Mexico. So welcome, Matt and Marcus on Zoom. If you want to say hello and maybe give a brief background, maybe as to what brought you to study microplastics.
Sure. I'm Matt. I'm a professor here in the College of Pharmacy at the University of New Mexico.
Hi, I'm Marcus Garcia, and I'm a postdoc here at the University of New Mexico.
I'm an inhalation toxicologist, really, and for decades I've studied diesel exhaust and coal combustion particles and all kinds of things. And over the last 10 to 15 years, we've been hearing more and more about microplastics. I was initially very skeptical, but we started trying things, and the more we scratched the surface, the more things became a little bit concerning. And so we've done some basic studies with cells and mice. We've been trying to understand what these things really look like in the environment and in our bodies. And so this is kind of a culmination. We have another study that we're going to publish soon. That's another kind of revelatory piece. I'm very excited about, but it won't have nearly the impact of talking about how much is in the brains. So,
Sorry, if you don't mind muting yourself if you're not talking. Thank you so much. Awesome. Thank you, Dr. Cameron. And then also, Marcus, maybe also what made you interested in microplastics?
Yeah, so kind of in the same context, like Matt said, few different things is I'm actually a pharmacist by training. So one of the things that really I like to focus on in my research is bringing that clinical component as well as the translational work with it. And again, just trying to see and understand what microplastics are doing in our bodies has been something very important. I originally started with a couple other publications because I have a big interest in reproductive health. So that's where we started to develop this method and start asking a lot of these questions. And as we've been going on, I mean, finding a lot of very insightful information and, really continuing to build and have better understanding even post this publication so we can bring that information to individuals within the communities as well as hopefully use that for some type of change, whether that be policy or better understanding overall.
and I'll add one other thing, which is a lot of our work with environmental health toxicology. We see overlaps between responses to environmental stressors like inhalation of ozone or wildfire smoke and changes that are being driven by the natural aging process. So being invited to be part of the longevity journal club is very cool. So I want to hear everybody's points of view on this.
Yeah, awesome. Thank you so much. Well, we're excited to have you. So as we before we move into the article, I just want to make sure we get people on the same page and what are we talking about when we mean microplastics? And also feel free to interrupt me if you want to kind of like add to what these are. But in looking at this, I was looking at both primary and secondary microplastics. Primary meaning manufactured to be small, um, and whereas secondary are as the environment breaks down, things like plastic bags and things like that, then you get these smaller micro and nanoplastics. And these can be found, um, you know, gi- given the advent of when plastics began to be massively used, they of course are accumulating in the environment because they don't break down. And so it makes sense that this is somewhat of a new area of study, given that, before the 1970s, that they didn't exist. Um, but certainly are becoming widespread within the environment. And as they break down, they can accumulate microplastics in the ocean, in landfills, possibly even the atmosphere. And so when we think about human exposure, this can predominantly come from the things that we eat, or like you said, inhalation. If it's coming from, you know, um, animals that have kind of also consumed microplastics. And then Mary found this list, this website, and we're going to send out the PowerPoint afterward, this plastic list, where you can kind of look up the amounts of plastics in different things run by a group out of UNC, I think, or yeah, UNC. And one of the surprising things on here was the amount of microplastics in rotisserie chicken, which, as we think about putting a hot chicken into a plastic bag, maybe that makes sense. Um, but and then kind of at the same time as this paper came out, another paper came out demonstrating being able to see microplastics using some imaging technology, where they kind of thought through possible further breakdown of plastics in mice and postulated about how this could impact brain health through occlusion or maybe causing additional inflammation. But of course, it's still, it's difficult to study and when it comes to correlation versus causation, which we'll get more into in the paper. So with that, if we want to dive right in the paper, unless there's any additional background that Matt or Marcus you'd like to add.
I so those websites or those sort of
Mhm.
lists tell you where you're most likely to be exposed and things like I just want to express sort of this higher level. We're getting it wrong because the industry around plastics has pushed this message of it's not our fault, it's yours. It's the fault of the individual for not recycling better or using too much of our plastic, and they should make better choices. And when we look at these lists, it's the same idea. Like, what can I do to improve my health? And I push back on those a lot, and I'm doing it more aggressively nowadays just because this needs to be elevated to federal legislation. I mean, not that any of us are going to do that, I get it. But we have a Clean Air Act that's 50 plus years old now that was very effective in making, the automobiles put catalytic converters on all the automobiles. So we've improved air quality and water quality through legislation and not through individual choices. And I just want to share that sort of thought idea that we need to be very cautious because the media plays into it and our physicians play into it. And it's like,
Mhm.
we really need to change the story here.
Mhm. Yeah, thank you. And as we get into the article, feel free to raise your hand and ask a question. And yeah, let's get right into it. So from the methodology, it seems like for both the 2016 cohort and the 2024 cohort of samples, the exact same sample collection protocol was utilized where small sections of organs were collected and this technique, pyrolysis GCMS, was used, where tissues could be chemically digested, and then that pellet was collected and could be compared to pure um, standards. And then looking across multiple tissues from this 2016 cohort and the 2024 cohort, there was a significant increase in the liver of just total microplastic concentration, and then a pretty high amount in the brain, which also was increased from 2016 to 2024. So when it kind of comes into PE, or polyethylene, that seemed to be the dominant amount. Was this surprising to you?
Not really, in the sense that,
Mm,
well, two things. One is polyethylene is probably the most widely used over the past 50 years plastic. The other thing is HDPE, high density polyethylene, has a high amount of crystalline regions, and this is where the polymers all kind of line up, and
mhm,
they have more durability.
mm.
And so we think that these crystalline regions are really part and parcel to what's surviving to the nanoplastics phase, and you'll get more into that a
Yeah,
little bit later.
yeah. One of the things that was kind of surprising to me were things that decreased from 2016 to 2024, like nylon 6, seems to be a pretty large decrease. I did see around 2015 there was a large plant that manufactures the precursor to nylon 6 that exploded and led to some global shortages. So I'm just curious what the hypothesis is around some of these still pretty widely used plastics that seem to decrease in the liver.
So keep in mind that some things were sort of artificially low in the 2016 groups because they weren't really at a level high enough to detect them very well.
Mhm.
And so when you look at the averages, you get these sort
Mm,
of like not PVC, for instance. PVC went up, and because those are percentages, that might be more of what's sort of crushing out the overall amount of
hmm
on there. So it's hard to take this data and make a leap to sort of other. Everybody wants there to be something related to COVID in this
Sure.
span from 2016 to 2024, and I just urge people, just don't spend your brain power. There's no way to sort that out from this data set.
Yeah. Yeah. I mean, it's with things like this, it's so hard to detangle correlation, causation, and yeah, it's yeah, very true. So yeah, like you said, polyethylene is one of the most widely used plastics in things like plastic bags and uh, predominantly used for packaging, and accounts for a pretty significant amount of the total plastics market. So then this is probably one of the most shocking, or I guess it's a pretty eye-catching um, uh, graph where you found that with in the brains of patients that have been diagnosed with dementia, there was this very high amount of microplastics compared to those within the same year, um, uh, where without dementia it was pretty low, across multiple studies, or across multiple cohorts, and this seemed to be increasing over time. Um, of course, I think that correlation versus causation is very difficult here too, because as I think this community well appreciates that, um, in patients with dementia, you have a leaky brain barrier, you have, even in possibly an increased lipid burden and things like that could ultimately promote microplastics in the brain. But yeah, if you want to talk a little bit about how you're thinking about why this might be, and any additional comments on this.
Sorry.
Oh, so I kind of agree. The blood-brain barrier is probably a key part of this, and you'll get to some images that sort of highlight like we didn't really see the refractile inclusions, which we believe are plastics. We didn't see them in and around blood vessels in normal cases, but they were really present in the dementia cases. And then the other issue is clearance is impaired in these samples, and then there's also atrophy in the brains of dementia patients. So it could simply be that at least part of that elevation is a reduction in the denominator, the overall brain mass, and they're just getting trapped in there. The inflammatory cells are very clearly playing a role in collecting them, whether they're collecting them just because they're there or whether they're a trigger for the inflammation. We're not ready to make a big stand on that.
Yeah.
I always make the silly comment. Dr. Alzheimer diagnosed his one and only dementia case in 1907. We didn't have plastics then, so we have to be very cautious in making that leap.
Yeah, absolutely. Yeah, I'm curious if there were any additional patient data that you were able to get on the dementia patients that might also add a clue. Like, I mean, looking at the standard deviation across the cases is pretty wide too. I'm curious if patients with particularly high amounts, were diagnosed with dementia, a while ago, or how long did they live with dementia, and did that in any way correlate with the load?
Yeah, we didn't look at that. That's a good question. We knew 6 of them were Alzheimer's, 3 were vascular dementia, and 3 were other. And in that breakdown, the vascular dementia were as 3 dots were the highest among. I mean, they weren't statistically different. So we decided just not to play into that discussion.
Hmm.
We put this out as a brief communication in part to make it simple, if
Hmm.
if that's
Yeah,
something you would. But we wanted it to. We didn't want to dive into a lot of the nuances of these things. Um,
totally.
so you're right. There's probably some aspects of the disease severity and manifestations across. I mean, dementia's got a lot of variability from case to case, so
Hmm. Hmm.
there could be something there.
Yeah. Yeah, so to get into some of the imaging, these particulates, being able to, see them with TEM and in the brain parenchyma is kind of shocking. Even though, like, sometimes I guess not co or coming from like not seeing a ton of these images, it's hard not to think like, oh, what that? So, is this like similar to what you would see in like a mouse treated with microplastics in the water, or um, you know, can you add a little bit more as to like if this was like kind of surprising to see, or how did you go about looking for this in the first place?
So in terms of that, we've
Hmm.
done some previous studies using mouse models where we actually it's a process called gavage. So ultimately, we put the plastic content directly into the stomach and then see how it's going to translocate to all these different organs. So we did that more from a metabolic standpoint was the initial test, but in that we did some visualization. And that was kind of what led us to start looking from a human perspective is because within the liver, kidney, as well as within the brain, from what we dosed, we did see some of these particles were translocating into these. So it gave us that initial idea to kind of take a look and then wanted to advance that further as we moved now looking with this study.
And one of the big issues, big is a funny word. These things were very small. What we
Mm-hmm.
saw in the liver and kidney appeared to be larger particles, and they seem to be agglomerated.
Mm-hmm.
Hmm.
Um, we're digging into this now. There's another little figure. I don't know if it's coming up where it's just the shrapnel that were isolated away from
Yeah.
the brain. We
Hmm.
think we think that in the brain those are really very dispersed. They're super small. They're less than 200 nanometers long. They're not really going to be visible on a light microscope.
Yeah.
Moreover, that's not what's being given in mouse studies.
Mhm.
People are using little microspheres. We used 5 micrometer microspheres, and we're able to show that they do get to the brain, not in great quantities, but they could appear in the brain after we digest everything away and show those, as Marcus was saying.
Mm.
Smaller particles, things less than 500 nanometers, are traveling there much more effectively.
Yep.
They get caught up, we believe, in sort of the enterocyte absorption of lipids in our diet. You form chylomicrons. We have data now that show that these things are getting into the chylomicrons, and that might be part of the delivery.
Mm.
But so the brain stands out as we really don't see the agglomerated larger particles. We just see these sort of individual things, and we're probably missing a lot of them
Mm.
as a result of them being so small.
Yeah, are there good mouse models for a deteriorated blood-brain barrier that you could use to try to see
For
if,
sure.
that has a big impact?
Yeah, in fact, we have now the Load 3 mice models from Jackson Labs. They're humanized tau, ApoE4, and A beta knock-ins. And we're doing both wildfire smoke studies as well as plastic studies coming up in the next year. Um, just as some tests, you can inject LPS and it'll open the blood-brain barrier. Traumatic brain injury models for mice. There's a whole lot of avenues to test that
Mm.
hypothesis, and there is a study that did that with. Oh no, it's a human study. They weren't using the best methods. They were using microscopic determinations rather than the mass spec. But they did show that there was a TBI related phenomenon of increased amount of plastics in the brain.
Oh,
Um,
interesting.
yeah,
Mm.
I can find that link and send it to the group later if you're interested.
Yeah, that would be great. Thank you.
Mhm.
Yeah, to kind
Yep.
of go on to the image that you mentioned before, using TEM after the chemical digestion of the brain, you find these um, particulates. Um, yeah, I mean, it's kind of crazy. It definitely makes me worried a little bit more so than I was before about microplastics. Um, but it's what are the controls that you can use for this sort of digestion? I guess coming from, being a wet bench scientist myself, I think, oh, yeah, I work with a lot of plastic. So can you comment on, the controls that you might use, negative controls, where you don't find this in the protocol that you use?
Sure. I, I think we've received more pushback on these images than anything I've ever done in my career. Um, so, so in the background, when we published this, we have had ready to submit another paper, and it's in preprint, and
Mm.
we're still wrestling with reviewers on it.
Yeah.
But we've done the blank images.
Mhm.
This is on a TEM grid. So if you look at the
Mhm.
like the little wavy, like the sort of semicircle things in the background, that's just part of the way that grid looks.
Mhm.
Um, so we can image that, and then we're using the solutions like potassium hydroxide, which is what we normally use. We used here. We're doing different digestion methods like nitric acid instead of a base. We're using hydrogen peroxide, which
Mhm.
is just pure oxidation, no change in pH. We still see these little shards, and the sort of coup de grace for our studies is coming out like this week. I'm literally we're working with a graduate student
Mhm.
from main campus, a biology student who's really interested in um, museum curation, right? And so she's got access to this repository of mammalian species collections. We have kangaroo rats from 1966 that were collected up in Rio Rancho, just north of Albuquerque, and we were able to go back out in May and collect 2025. And I can tell you, it is night and day. There are
Mhm.
no little plastic shards like this in 1966 samples in brain. There's all of them, just like
2025.
lots of them in 2025, and they're just wild roaming around in the little
Mm.
dry river beds that we have here.
Mhm.
Um,
Mm.
And I was just going to add just kind of why the importance of that in terms of why we're looking at 1966 and 2025, and also why we chose to use these kangaroo rats is you did a great job of showing that image, showing how plastics accumulation in the environment has gone up since the 1950s. Well, it's based off of that, and we've done some initial mammalian studies to kind of mimic that same kind of accumulation. So we did a study beforehand where we took some field mice that were collected in the 1970s, and then utilizing the technology around PyGCMS, all 5 of those samples that we tested had no plastics present based off of that analytical technique. As early as 1974 is when we saw 2 out of the 5 start to show a little bit of that plastic accumulation. So based off of that threshold, we wanted to decide, okay, well, if we can get tissues even earlier than that and kind of look at that from a visualization perspective to answer this question, that's where we're kind of able to establish that what is the true control of showing any differences between plastic, because we know that they're so ubiquitous within the environment and then within us at this point that we want to try and establish that.
Yeah, it's really interesting. It makes me think about how the potential for heterogeneity of, wild mice samples in different locations. Is that anything that you're exploring, like trying to get wild mice from places that might be have less exposure to plastics?
Yeah. We, we, we don't really have funding for this.
Yeah, that's fair.
We've got, we have a lot of funding actually by National Institute of Aging to look at wildfire smoke.
Hmm.
So yeah, there's a lot of opportunity. I think the geospatial question is the biggest, especially from the standpoint of a lot of our food comes from agricultural practices that require irrigation. I would like to look at equatorial regions where they just get rainwater, and the rainwater should be cleaner. It doesn't have contamination from as much contamination from human experience and waste like landfills and stuff. So I'd love to do some studies like that, but it's sort of outside of. At the end of the day, I get funding from NIH. I'm not going to get it from NSF. I am what I am. But these are the kinds of important research questions that need to be looked at.
Yeah. Yes, try.
Yeah, I have a follow-up question to what you just said. Um, Also super interested in microplastics. Just for some background, I am a physicist, but working in the biotech longevity space. So I'm approaching this question more from a point of, can we, use modern technologies to solve this problem? Like how do we break down microplastics and or maybe create materials that are more, biodegradable. So that's mostly my approach. But I'm super interested in what you just said, which is, I mean, coming from India, and I've seen like, people like I've grown up on agricultural lands, and I've seen like these practices being done. So like it's true that most of these practices depend heavily on like rainwater, but like it's also true that a lot of the plastic waste from the West gets uh transported to the countries where most of these agricultural practices occur, and then there's a lot of like the pipelines of distribution of water and storage of water. They're not the best. They could be even like this could be introducing more contamination than less. I don't know. So I'm wondering if you've thought about those confounding factors, or if all that is super vague at this point, and you're trying to decide on more direct lines of research.
So all of the above are potential contributors. You're right, and um, I, I think part of where I have an unconventional viewpoint on this plastic story is that I'm seeing a story that originated from oceanography. Richard Thompson published in 2004. He was sailing across the seas, and he's like, "Wow, there's a lot of plastics and a lot of microplastics, and is this what's happening?" And the story exploded from the oceanographic point of view and then broader ecological. But a that's not a pathway of exposure for me. And maybe it's because I live in New Mexico. I'm at 5,000 feet altitude in a landlocked state. I'm not exposed to ocean. I mean, we get seafood, but it's trivial compared to the grains that are coming from Iowa and Nebraska and things like that. So, I'm trying to think. I hear hoofbeats. I'm gonna focus on horses first before I go to zebras. Um, the bulk of waste in this country is still buried in the ground in this country. We do ship stuff overseas. There was a fantastic editorial in the New York Times like 4 months ago that said the story you've been told about recycling is a lie. And he talks about how, where's my prop bottle of water, right? Okay, so this bottle of water, this plastic bottle started in the sands of Saudi Arabia, right? It was pulled out as crude oil. It was shipped maybe to Louisiana in the region we call the Cancer Alley, and it was refined into ethylene and then into polyethylene, and then it was created into a bottle. And you think about like the months or years it takes to do this. It goes into my hand. I drink it in 30 seconds. This bottle means like nothing to me in my life, and then I dutifully recycle it, and then it goes on this other journey, ultimately being shipped over to Bangladesh, where it might be incinerated or thrown back in the ocean. Like, what an absurd concept for a thing that means absolutely nothing to my existence. So that article like really framed exactly what you're saying. Like, there's so much wrong globally about what we're doing in these plastics, and maybe the solution is old technology. What can we just say this instead of being a single-use plastic? We now incinerate it and turn it into energy, and it's a dual-use plastic. You know, there might be simpler things that we're afraid of doing. I don't know.
That is certainly my view. I think there are existing solutions that have existed for centuries that people are just, they refuse to use because it's maybe 100 of, it uses 100 more inconvenience to them or something. So yeah.
Yeah. And
We also,
and go ahead. I'll say my field of ventilation toxicologists might be part of the problem because waste to energy requires incineration, and it can be done much more cleanly than coal combustion for energy. It's more efficient than coal combustion for energy, but everybody's afraid of incineration because it's going to create pollution. Like not that much is really the answer. So there's problems.
Mm-hmm. We also have a question from the chat from Brandon Kaplan, who asked, who said, thank you for this research. Has your work crossed over at all with research on how to remove plastics from the body?
Oh my. Did you see the story about Orlando Bloom this week?
No.
Okay, well, everybody's favorite elf apparently paid over 10,000 euros for therapeutic plasma exchange. So this is coming at us. Both the media is asking my opinion about whether apheresis can be used to get rid of plastics, but also the apheresis companies, therapeutic plasma exchange companies, are coming to us and asking for us to be advisors or potentially run samples for them. Mm-hmm. And what I'll say is, yeah, it probably works, but I mean, think about the island of Manhattan, 10 million people thereabouts. Not many of them are on the subway at any one time. And in fact, every night the subway empties. And then it fills back up pretty quickly. So you can pull these things out of blood, but your diet is probably just going to put them back in within a matter of days or weeks. And it's not going to really change what's in the brain or kidneys or liver. The other ideas that we've heard about are like, what if we engineer some sort of bacteria that can go in the gut and digest these? Wow, are we really going to do that? Are we going to start putting strange bacteria in our guts and hope that works out without dysentery or IBD or some autoimmune thing? somebody brought up the idea of Olestra, and it's like, if you even remember Olestra from back in the 80s, we made potato chips that were, going to inhibit fat in uptake in the gut, and people just had rectal bleeding and
Yeah. Or another thing that's been brought up is around zeolites. So this is just like a mineral. So ultimately, the idea is creating a spray that you would ingest the same time you're eating some food, and then technically the way it's been pronounced or been talked about is that it binds to these plastics and allows them to flow through. I mean, there's so many different mechanisms where that is very tricky and challenging to even say that that would be a probability, but we're seeing a lot of these products ending up on the market. And I think we need to see a lot more research based around them and be able to answer a lot of those definitive questions, because there's a lot of caveats, I feel, in terms of like how those actually bind or how they're working as well.
That's interesting. Thank you, Bonnie.
We've got another question in the chat, Rebecca.
Yeah, if you want to read that, Mary.
Sure. It looks like it comes from Jonathan Berman. He says, what do you see in kidneys, I think? Is there a decline in GFR in mice?
So we've only started. We finally got a good kidney physiologist, pharmacist involved in our studies, and we're gonna study that a little better. Mm-hmm. What I'll say is, in this paper, we have some nice images that highlight just the anatomy of where we think plastics are falling in the kidney, and it's clearly focused in the glomerulus. So they seem to be going where they should go and getting filtered where they should get filtered. Mm-hmm. And then we saw a trail that looks like it's going down the distal tubule. So when we think hard about what the regions we see these deposited in kidney and liver, I think the kidney and liver are doing their job. We have a lot. We haven't even looked at GFR. The question I always ask about any given disease like CKD or dementia or MS or anything, is it going up in incidence in our world? And so I haven't thought about, I don't know if CKD is going up as an independent thing that isn't a function of aging, right?
Yeah, really interesting. Yeah, and then kind of moving forward in the paper, we found you showed that in dementia samples you see a lot more of these refractile inclusions, which are these structures that are kind of difficult to visualize. These very bright spots where electron beams appear to appear pretty distinct from the background and bright. Do you want to talk a little bit more about what these refractile inclusions are and as they appear with the imaging?
Yeah, we think there are certain kinds of plastics, and in particular, we think that they are the crystalline regions of, say, high density polyethylene or polypropylene. Obviously, wouldn't be styrene butadiene rubber, which comes from tire wear. So this isn't a universal detection, and it could also be that there are other crystals in the body that are formed, like oxalates, that are creating these. So we have to be cautious. But what I like about especially the lower panel is you clearly see these little refractile inclusions in the blood vessel. You don't see them in the blood. There's all these little red blood cells in there, and you can scan all you want, and you won't find one. And it comes back to people's interest in trying to find these in the blood or take them out of the blood. It's pretty low yield there. And then in the immune cells above, and maybe I can take over the screen share later at the end. I'll show you like that overlapped with a raw H&E stain, and it's really cool because the things that light up as refractile are not all the inclusions in the macrophages. So there's definitely some dark inclusions that maybe it's old dead and dying cells or soot particles or something like that. And then adjacent to that or in other areas, you'll see these things light up. So it's a really cool method. This is polarization wave microscopy. it's been eye-opening, but at the same time, it's not like a chemical signature because these things are just too small for Raman or FTIR imaging.
Hmm. Hmm. That's really interesting. Yeah, I'm happy to switch over and you can share your screen real quick if you want to show that.
Yeah, let me see if I've got that slide prepped. Let's see. So,
No
okay,
worries.
so
If not.
here we go. So if you stop sharing,
Yeah.
and I can do a quick share.
And then if you don't
Hopefully,
mind muting when you're not asking a question, that would be great. Thank you. Yeah.
okay. So this is just the H&E, and you can see these macrophages with dark inclusions, and I'm just going to toggle back and forth between the overload thing. So you see particles show up where you didn't even realize there was a particle. Um, it's just a really cool and informative way of looking at this that sort of better than previous methods that we've had, and definitely lets us look at smaller things than you can, with conventional spectroscopic microscopy that's been used for plastics identification.
Yeah, that's really striking. There we go. This back up. All right, and then kind of with this in thinking about possible links to actual possible causative versus correlative links. I think from your paper, you make it clear that this is an observation. You're not trying to make a causative claim, but there is a critical need to better understand the potential consequences to microplastics accumulating in the brain and that the hallmarks of dementia, like this blood-brain barrier integrity or itself, certainly play a role. So then in thinking about, I know Marion, Vishal, and I talked a lot about the things that we love is like really bringing awareness to this is something that I had not really thought a lot about. And then, there's still so much to know. And I think that we kept on coming back to that throughout the discussion that, the mechanism of how it's getting there, but also, how bad is it when it gets there? How causative is it? Is it, making dementia cases worse or leading to directly leading or being causative to pathophysiology is still really, really left unknown. And I think we talked a little bit about the critiques along the way. And if anybody has a comment or question about either a critique or things that they liked or unknown, feel free to raise your hand. And then kind of going into the limitations, of course, it's such a new area. So there are certainly a number of limitations, whether that's even limitations that may result into even underestimating the amount of microplastics in the brain, whether this is, because you're comparing to a very pure standard, so the amount of microplastics quantified ends up being less than what is actually there, or perhaps because during the collection or given that you're collecting brains postmortem, you might also lose microplastics in the CSF or other things like that. And then kind of limitations on the other side, you note a 25% within sample coefficient of variation, and the potential for lipids and other things like that causing problems. So be interesting to hear kind of how you're thinking about the future of getting over some of these limitations and what you think the future is to studying microplastics in different organs and even the brain.
Oh yeah, absolutely. And I think you have a lot of key points with this, and especially one thing to consider is around the lipid interference. So with that, polyethylene specifically and lipids have the same look in terms of how those polymer chains are formed. And then especially as you do the combustion with PyGCMS, as it's cut down, it's cutting down those chains, like that can cause some interferences with that. So some of the things that we've been doing in terms is looking at additional digestions in terms of once we isolate the pellet, whatever lipid is remaining in that, we do use some organic solvents, more specifically cyclohexane, to be able to remove that, as well as now that we know that a lot of these plastic particulates are roughly around 200 nanometers in size. We also incorporate a filtration system where we're not only filtering to capture anything larger than 50 nanometers in size, but with the cyclohexane, it's ultimately washing that lipid through that filter as well in the process. So those are some of the things that we're looking at, as well as really going deep into deeper dives into the chromatograms that are formed from the pyrolysis system. So there's interesting ways in terms of like the peak formations where we can actually look and decipher, is this actually polymer in structure or is this lipid in structure? So being able to work through a lot of those to be able to identify.
Yeah. And so the kind of big thing that's out there is we took these samples from the frontal cortex and we extrapolated the total mass to the rest of the brain, and the plastic spoon analogy has been unavoidable in the media. That's overestimated for a couple of reasons. One is the lipid, and Marcus is now looking and mapping an entire right hemisphere of a human, and the frontal cortex is by far the highest. So it's kind of like estimating the concentration or like the population of this country by taking a random population density of Manhattan, right? So yeah, the rest of the brain seems to be lower. We do have this fat problem in that plastics move to fats. And so we see the highest concentrations in the myelinated region. Okay, that's great. With the cyclohexane wash, we can pull that down a little bit, but it doesn't go down that much. And we're getting rid of the lipid and we're improving our signal. So that is just a part problem that we have going forward is the amount, the fatty regions or the lipid rich regions is going to be higher, and people are always going to be arguing with us about interference on the PyGCMS. So, and maybe the bigger picture is this isn't like some fundamental question of physics, like the speed of light. We don't necessarily need to know some absolute value of what's in the human brain. We just need a marker that we can use consistently across populations to start saying, okay, population without dementia or population with dementia, and then make head-to-head comparisons with a standardized method. So we're trying to be conscientious of the analytical chemistry, but also of the need for epidemiological research in this area.
Yeah, absolutely. That's so interesting. Mary, if you want to go ahead and move forward on the slide. Sorry, I had a computer issue, so we switched out real quick. You can go forward one more time too. Yeah, I think we hit a number of the future research areas during the discussion. Thinking about how micro nanoplastics are impacting not just dementia, but I think you also talked about TBI and some of the other pathophysiology that could be impacted by micro nanoplastics in brains. And then also thinking about modes of exposure, but I really appreciate your comment about, it's not just on the end user to fix, how we expose ourselves, but also globally, governments and people who are able to, impact what exposure methods are allowed and further techniques. But are there any additional research that you're particularly excited about?
So you sort of faded out there. Say that again.
Are there any additional future areas of research that you're particularly excited about, whether in your lab or other labs working on this?
I know it's a minefield, but autism is very interesting to us because we know that they're in the fetal brain. Um, Reproductive effects. We've got very clear and concerning data related to sperm motility and related to preterm birth and areas like that. So it's fun for us because we just get to be the plastics people, and other groups come to us with their expertise in their fields. So yeah, every day brings something new.
Oh yeah, that's awesome. I love the idea of being a bridge and enabling research to be done in these areas. Yeah, Mary, if you want to go ahead and move forward a slide. Yeah, we would love to just open it up to anybody within the meeting who wants to maybe pitch how they're trying to minimize their exposure while hoping for the powers of that be to help minimize global exposure as well. But are there any things that people are doing that they think might be helping to minimize their exposure, or also Matt and Marcus, are there things that you do now to help try to minimize your exposure?
I see a hand raised from Georges, if you want to go ahead.
Yeah, thanks, Mary. Actually, yeah, that was exactly the question that I had on the exposure mechanism. So far, my idea was roughly it's from food contact applications, so the plastic bottle I'm drinking from or whatever, any dairy products in plastic containers, stuff like that. But based on these animal results, it seems like that the environment is broadly contaminated. So no matter what or in which container I buy from in the grocery store, it won't make a big difference. So what's your estimate on this? Like if you were to give a share, like 80% is broad environmental, 20% is direct food contact source or so. What's your feeling there right now? So where is the main exposure coming from?
That's a great segue. If I can share screen again. So I think most of it is from our food. I don't think it's from the air we breathe. I know a lot of people get worried about that, but less than 1% of the total particulates in the air, according to our calculations, are plastic. So it's still a very low amount that we're breathing. The food is interesting, and oh dang, where did it go? Hold on a second. Oh yeah. Okay. So share. And I can make this full screen. So we've got high school students that come over the summers and undergrads that do these projects. And so this is actually my kid, along with John Boyle and Giselle Sanchez. Giselle's back this summer. This is what they did last summer. So the percentage above each of these bars is the percent fat that's in each of these products. And you can see this is polypropylene only, but venison, grass-fed beef, chicken, ham, bacon. Those are all really low. They're not processed foods. But then you get to pepperoni, beef jerky, and vegan burger. They're magnitudes greater amounts. This is on a log scale. And to the point about fat, we included in here whale blubber. I forgot to include, we have permission and licenses to have analyzed whale blubber. But it's 80% fat, but it's only sitting here right in the middle, about 50 micrograms per gram, much lower than in kidneys. So we really think that man, I do not like what's going on in the world with federal science policy right now. But the whole like MAHA emphasis on processed foods and microplastics, that has merit. I don't think it means we should stop measles vaccinations by any stretch, but there's something to this.
Yeah, that's
So I
as someone who eats a lot of vegetarian and vegan burgers, I'm like, ah.
right. So we're this summer with our high school students, we'll be looking at vegetables and trying to sort this out. And hopefully we'll have a paper that comes out in the fall that puts all of this together, because obviously what's happened, the recommendations that are out there are based on methods that don't capture what actually absorbs into our body, because they're looking at large particles. 5 micrometers is sort of the lower limit that FTIR and Raman spectroscopy can handle. And so most of the data that's been put together is irrelevant for our existence. So we're hoping to get this out there soon.
Yeah, well, I'm gonna really look forward to seeing that paper for sure. In the last 2 minutes, Mary, do you want to move forward to the slide to show some of the companies that are thinking about this?
Yeah. So we also have one question from, I think, Edna, if you want to unmute and go ahead while I pull up the slide.
Wonderful, Mary. Thank you so very much. Matt and Marcus, thank you for this wonderful presentation. If this question came up, apologies for duplicating. I am a pharmacogenomics and longevity pharmacist. And my question is really about the impact of this from clothing and topical products like skincare products. Do we have any data on that?
So in terms of skincare products, we don't have much data on that. However, one of our other lab members, and actually she's a research assistant professor, did an extensive job last summer in terms of looking at baby clothing. So looking at fibers and what they're able to extract and analyze. I believe that she's in the last phases of getting that ready for publication, but there's been a lot of pretty interesting data that she was able to collect around that, and predominantly like the polyester fibers is what she was able to collect, and then do some mouse studies to look at how that's affecting from not only a inhalation but also a GI standpoint as well.
That's super interesting. And then Mary, do you want to, in the last minute, walk through some of the companies that you found to be working in this space?
Sure,
Yeah,
I think
also happy to get some comments from Matt and Marcus on what you think about some of the companies in this space too.
Yeah, absolutely. I think Matt and Marcus, if you want to chime in here just in the interest of time, I think, yeah, Rebecca and I were chatting and take on sort of in industry, especially being marketed direct to consumer.
Yeah, it's the Wild West now. This is going to happen. It's I'm not familiar with all of these, but like Blue Land and the Blueprint, and they've got these test kits. They will give you a number, and so you will pay a couple $100, and it will tell you have some. But there's no clinical correlate. It doesn't tell you how much at risk you are for anything.
Yeah.
So they're also using an FTIR type approach. So it's going to be larger plastics, and it's not necessarily going to be the nanoscale stuff that we see embedded in our brains.
Yeah.
For the Clarify Clinic and these, I actually think there's really compelling, cool stuff around plasma apheresis and longevity. The senescence associated secretory profiles and that whole thing, like a really cool area of research right now. And there's parallels between that and what we see when we have our lungs exposed to like wildfire smoke. There's shedding of peptides that kind of overlap a little bit with this SAS profiles. There's a lot of neat stuff in that space. So plasma apheresis, I don't want to throw the baby out with the bathwater. There might be stuff there that we can explore. Does it reduce plastics from before to after in the serum? Yeah, probably. Does that make a heck of a lot of difference to our health? No, I seriously doubt it.
And I would even say, like, in terms of blood testing, even looking at what we've been able to show in the publications, that we're not really seeing too much plastics, at least in terms of the blood that we're seeing within the brain itself. I mean, a lot of times it's really dependent on times in which we're eating the food, because that's when it's going to be causing more of that transient effect. So it may not stay at a certain point. So then down the line, depending on when you're testing, you may have a higher concentration within the blood at one point versus the other. So it's also like trying to be able to figure out that definitive answer around that.
And there's so much about this field that we just don't know.
Yep.
So maybe.
Yeah. Well, thank you so much. And that kind of takes us to the end of our conversation. I know we went slightly over. Thank you so much to people who hung around. And we will be making sure that these slides and the recording get shared within the community of people who weren't able to make it. And thank you so, so much, Dr. Kim and Dr. Garcia, for making the time. This was an awesome discussion and definitely a field that I'm going to pay more attention to going forward. So thank you for doing such great work.
Well, thanks for having us.
Yeah, thank you guys.
All right. Have a good weekend.
Yeah, you too.